My name is Mancia, and this is my microcosm model presentation for ESVP 416. Here's the overview. So we do models because they're small-scale lab-based and they tested theory. The benefit of modeling is that it saves time, resources, and cost because they are on a smaller scale. Some of the pitfalls and limitations are that it assumes concrete values, it simplifies nature, such as not including birth rates, death rates, or migration, and it skews data. So this is my first model that I chose to do is estimating population size. So this model allows scientists to find the number of organisms in a population without having to count every individual within that population. And the parameters for this one were population size, pond size, and net size, and this was estimated using the mark and recapture method. For each run, I used the different size of pond, population, and net. The first one being large, second being medium, and the third being small. The fish were caught and marked after each run, and then they were returned to the pond. They were caught a second time, and the amount caught and the amount that were previously caught and marked were used to estimate the population size using the Lincoln-Peterson estimate, which is the total amount in the traps times the total in the traps caught the second time, divided by the amount caught the second time that were previously marked. So in this model, the organisms seem to be very evenly concentrated in large population sizes and in a large pond compared to a smaller pond with a small population size. It seemed that the smaller population was more spread out and dispersed, making it difficult to catch and mark organisms and then recatch them. So this model could be used in a real scenario with an actual pond and fish population because it allows scientists to sample smaller sizes and not have to look at a larger population. I think the data was skewed for the smaller population because it is difficult to recapture marked organisms. Some of the pros are it can save time and effort. It's better for large populations. And the cons are as assumed population is closed. For the second model, I choose a mark and recapture the same as the first one. The parameters are population size, distribution, and trap experience. And this one is again using the Lincoln and Peterson estimate. So each run had the same population size, trap time, the same amount of traps, and the same distribution, but they each had a different experience. The first one was a neutral experience, the second was a negative, and the third one was a positive experience. Again, this is using the Lincoln-Peterson estimate, where it's total on traps times total on traps the second time divided by the amount caught the second time that were previously marked. The second and third runs were the most accurate when the trap experience was negative and positive. I expected that these runs for the negative and positive traps, that the data would be skewed and it wouldn't be accurate because of the excessive amount of voles returning to the traps with the positive experience and the insufficient amount of voles returning to the traps with the negative experience. For the third one, I chose the microcosm community ecology model. This model tests the interaction between species in a community and explores competitive interactions and predator-prey interactions. The parameters for this one were volume, light, bacteria population, paramecium population, and didinium population. For each one, the volume and light stayed the same. The bacteria was increased. For the first run, the Buceria died off, so I increased the bacteria for the second run and decreased the didinium. The more bacteria increased helped the chance of survival of the paramecium. And then I had to kind of manipulate all the data just so that the Buceria wouldn't die off again, because it does have a slower growth rate than the P. aurelia. So you can use real microcosm data on population sizes, resource levels, and environmental conditions, and then you can compare those patterns to model predictions, and it demonstrates the competitive exclusion principle and suggests long-term coexistence. Usually requires some degree of niche or segregation, and then there's a competition of resources, as we've seen in the P. aurelia and P. buceria, because they have the same, or they thrive off the same resources, but P. aurelia grows faster and can repopulate faster. The pros are: can save time and effort, it's cost effective, it has control of the variables, but con is that it does not take into account other natural factors. The next model I chose to do is honeybee foraging. This model simulates foraging behavior of honeybees. It illustrates the Charnov's marginal value theorem, and it predicts. So for the each run, the B1 forage time and B2 forage time stay the same, but the flower distance, nectar amount, and extraction rate changed. The increased flower distance and nectar amount increased the collection rate of both bees, with the bee with the longer foraging rate had a smaller collection rate. So this demonstrates that the bees that have a longer foraging time doesn't necessarily mean that they collect more, and they could be wasting optimal time by staying too long at one flower instead of moving on and getting more nectar. Pros is that it can save time and effort as a controlled parameters, and it is repeatable. Con is that it's unable to modify plant distance for the bees separately and is not taking consideration the bee behavioral patterns. For the next one, it's the dispersal corridors. This model simulates dispersal corridors and movement of animals. Animals, they need to move to find food, to find a mate, or to find breeding grounds. And when a habitat is disturbed, animals with specific habitat needs become isolated, and the parameters are remaining habitats and number of moves for an organism. So for each run, the remaining habitat was decreased, and the number of moves, which I kind of understood as like the amount of energy. So for increased fragmentation and decreased habitats, increased the difficulty of reaching the breeding pond, also increased. And then so for the environment with the most habitats remaining, the frog was able to make it in 30 moves, but with 40 habitats, it made it in 40 moves. So it wasted more energy, and the frog with the habitat with 25 habitats, it didn't make it to its location. So it made it challenging for the frog to be able to go to its breeding area. The next one is estimate stream diversity. It explores the effects of water quality on a stream's biodiversity, and the parameters are amount of pollution and sampling time. So for each run, the sampling time remained the same, but for run 1, there was no pollution. For run 2, it was moderate pollution, and for run 3, it was severe pollution. From no pollution to moderate pollution, there's not much change in the biodiversity of the stream, but the stream with the moderate pollution began to reach its peak species amount quicker than the stream with the no pollution. So no pollution stream and the moderate pollution stream likely had the same number of species, because the increase in pollution tolerant organisms in the moderate pollution stream evens out the other organisms that were lost to pollution. So one of the causes, it does not distinguish between pollution tolerant and pollution intolerant organisms, although it does include it in the model. And for the next one is island biogeography, which simulates MacArthur-Wilson's island biogeography equilibrium paper, and it shows the organisms on islands have a higher extinction rate, and the parameters are island distance, island diameter, taxon, and migration rate and mortality. So for run 1, the species average was higher on island 2 than on island 1, even though they had the same parameters. For run 2, the species average on island 1 was higher than island 2, which was larger and farther away. For run 3, the species average on island 1 was higher than island 2, which moved farther away but grew larger in size. So this model has been used in a real experiment with like mini islands made from mangrove roots in the Florida Keys. In the real experiment, the larger island had more species, and this is not reflected in this model. I think it was easier for organisms to migrate to the smaller island nearby than the larger island farther away, even though it's a lot bigger. Some of the pros are it can save time and effort, it's cost effective, you can manipulate mortality and migration rate, and the cons is that it does not account for birth rates. And then for the next one, the fragmentation model. When considering habitats such as a patch of forest, it's important to understand that the patch is not uniform in how it is utilized by species. Different species are found typically deep within the forest in the interior, and the parameters for this one were remaining habitat. So for each run, the interior percentage decreased. The increased fragmentation decreased the amount of area for an animal to live. It prevents the animals from having habitats, as they are usually found in the interior of a forest and not on the edge. The forest with the remaining habitat of 50 has minimal area for an organism to thrive in. So this model doesn't test the effects of fragmentation on any specific animal, but it shows the amount of available space to an animal. This model can be used in a forest because it only tests the interior and edge percentage. And to test any theories about fragmentation effects on animals, forests with a high and low interior percentage can be identified and then compared to see if there's a difference in the amount of animals present. Some of the pros for this type of model are that it saves time and effort, and it's a simple demonstration of how fragmentation works. One of the cons is it does not show the effects of population. And here's the sources.